The leaves are finger-length and feathered, and so green they look almost artificial in the early light. A woman named Priya Mehta, representative of tens of millions of farming families across Rajasthan and Uttar Pradesh, strips a handful from a low branch before the sun has fully cleared the horizon. She does not weigh them. She does not look at them. Her hands know exactly how many. Her grandmother taught her. Her grandmother's grandmother taught her grandmother. Neither of them would have called this medicine. They would have called it breakfast. The National Institutes of Health has now published over 1,300 peer-reviewed studies on the tree those leaves come from. Welcome to the vault. That is not a metaphor. There is a documented body of scientific literature, institutional, funded, published in indexed journals, that describes a single perennial tree as a source of complete protein exceeding beef by dry weight, a natural anti-inflammatory, a documented hypoglycemic agent, an antibiotic active against drug-resistant bacterial strains, and a water purification mechanism effective at 99.99% bacterial reduction. That literature exists. What does not exist in any Western nutritional guidance framework is a single government-issued recommendation to grow it. That gap is what this file is about. Think about what you spent last week on protein. Ground beef at current retail averages $6.50 per pound in the United States. A mid-range whey protein powder runs approximately $1.20 per serving for 25 grams of protein. A dried Moringa oleifera leaf, gram for gram, contains more usable protein than beef, more calcium than milk, more iron than spinach, and more vitamin A than carrots. On measurements confirmed by the USDA's own nutritional database, filed and available, not disputed. If you are managing blood sugar with a prescription, the average monthly out-of-pocket cost in the US sits between $35 and $90, depending on your coverage tier. A 2009 paper in the Journal of Ethnopharmacology documented the hypoglycemic mechanism of niacinamide, a compound found in moringa leaf, in controlled animal studies. No human trial followed. You kept paying. The tree kept growing in countries where people could not afford not to use it. This is not ancient history. The timeline is recent enough that people who made these decisions are still alive. In the 1940s, British colonial agricultural policy across the Indian subcontinent issued land productivity classifications that formally designated traditional food trees, including Moringa oleifera, as non-commercial. The designation was not botanical. It was economic. Trees that could not be taxed through a commodity supply chain were trees that did not count. The classification shaped which crops received colonial agricultural investment for the next 2 decades and left a legacy in post-independence extension farming programs that persisted well into the 1970s. In 1979, the WHO published its first essential medicines list. Compounds documented from moringa, including several with established anti-inflammatory and antimicrobial properties, were present in the ethnobotanical record the WHO drew from. The tree itself did not appear on any recommended crop promotion list. The synthetic versions of several comparable compounds did. In 1997, the United States Department of Agriculture's Nutrient Database published a formal analysis of Moringa oleifera leaf confirming its protein density, amino acid profile, and micronutrient concentration. The analysis was accurate. It was filed. A proposal for follow-up human bioavailability trials was submitted through the standard federal research funding pathway. That same fiscal year, the U.S. soy protein isolate market crossed $4 billion in annual revenue. The follow-up trials were not funded. In 2006, the National Institutes of Health began indexing a growing body of literature on moringa isothiocyanates, specifically the compound MIC1, then documented in its anti-inflammatory mechanism at the Johns Hopkins Bloomberg School of Public Health. A 2012 paper in the journal PLOS1 confirmed the mechanism operated through the NRF2 pathway, the same cellular pathway targeted by several patented anti-inflammatory drugs currently generating combined annual revenues exceeding $20 billion. No FDA investigational new drug pathway was opened for MIC1 in its natural plant-derived form. The patents on isolated synthetic analogs, however, These were filed. Now look at what the tree actually does. In documented nutritional analysis, gram for gram, moringa leaf contains 25% protein by dry weight. Beef contains 26%. The difference is 1 percentage point. The difference in cost of production is the difference between a perennial tree and an industrial feedlot. The protein is complete. All 9 essential amino acids are present in moringa leaf in proportions consistent with human dietary requirements, confirmed in analysis published by the Food and Agriculture Organization of the United Nations in 2011. The calcium content is 4 times that of milk by equivalent dry weight. This is not a folk claim. It is a figure in the USDA Nutrient Database, entry 11,975, available to anyone with an internet connection and 30 seconds. 7 distinct glucosinolates have been isolated from moringa leaf and seed. Glucosinolates are the same compound class under active NCI investigation for their role in cellular protective mechanisms. The National Cancer Institute's plant-based compound screening program logged moringa-derived glucosinolates in 2003. No clinical trial arm was opened. The compound niacinamide present in moringa leaf demonstrated statistically significant blood glucose regulation in peer-reviewed animal models. The mechanism involves pancreatic beta cell protection, the same cellular pathway that is the target of several second-generation diabetes drugs currently in phase 3 trials at a combined development cost exceeding $800 million. Moringa seed extract demonstrates documented antibiotic activity against Staphylococcus aureus, including methicillin-resistant strains in studies published in the Asian-Pacific Journal of Tropical Biomedicine in 2012. No pharmaceutical development pathway followed. The seed powder, when introduced to turbid water at a concentration of 30 to 150 milligrams per liter, produces coagulation flocculation that removes bacterial contamination at a documented rate of 99.99%. This mechanism was formally acknowledged by the World Health Organization in a 2010 technical brief on household water treatment. It has never been incorporated into a single WHO-recommended municipal water treatment protocol. The global water purification market was valued at $29 billion in 2010. Now follow the money with precision, not inference. Between 2000 and 2015, 47 patents were filed internationally on compounds isolated from Moringa oleifera. The filing entities include subsidiaries of major pharmaceutical holding companies, several of which are publicly traded and whose annual reports are available through SEC filings. The patents describe novel extraction methods, isolated compound formulations, and synthetic analogs of naturally occurring moringa constituents. Of those 47 patents, 0 have progressed to a commercially available drug product as of the date of this recording. A patent without a product is a legal instrument. It prevents competitors from developing the natural compound along similar lines, while the patent holder decides whether commercial development serves their existing revenue model. This is not speculation. It is the documented function of a defensive patent in pharmaceutical portfolio strategy, described in exactly those terms in industry white papers published by the International Federation of Pharmaceutical Manufacturers and Associations. The soy protein industry spent $3.2 million in documented federal lobbying between 2010 and 2020 across USDA and FDA budget allocation processes. The moringa industry, which is to say, and the network of small farmers and NGOs attempting to introduce it into Western nutritional frameworks, spent, by available record, effectively nothing. Budget allocations for alternative protein crop research followed accordingly. 47 patents, 0 drugs, millions of lobbying dollars defending incumbent proteins, and a tree that anyone can grow, producing compounds that several of those patents acknowledge are biologically active. If the compound works, and the patent confirms it does, what is the financial logic of stopping before phase 3? That is not an accusation. It is a question the filing documents raise themselves. Here is what you can actually do today, with what you now know. Moringa oleifera seed is legally available for purchase and cultivation in all 50 US states throughout the European Union and across the UK, Australia, Canada, and New Zealand. It is not a controlled substance. It is not a restricted agricultural import. It is a tree. In USDA hardiness zones 9 through 11, Southern California, Arizona, Texas, Florida, Hawaii, it grows outdoors year-round as a perennial. In colder zones, it grows vigorously in containers and can be kept as a cut-and-come-again plant, harvested for leaf before overwintering indoors. Plant seed directly into well-draining soil or a container mix with good aeration. Depth: ½ inch. Germination occurs in 5 to 12 days at soil temperatures above 65 degrees Fahrenheit. The plant grows fast, faster than almost any food tree in temperate container cultivation. First harvestable leaf in 60 to 90 days. In 90 days, you will have more usable protein per square foot than any crop in the USDA's recommended backyard planting guide. Dried leaf powder is commercially available from reputable suppliers in the interim. Look for organic certification and third-party heavy metal testing on the certificate of analysis. The powder concentrates everything in the leaf, including any soil contaminants, so sourcing quality matters. 1 teaspoon of dried powder daily is the most common documented supplementation level in nutritional studies. As with any nutritional change, and this constitutes a significant one, discuss it with a qualified practitioner before incorporating it, in particularly if you are managing blood sugar levels or taking cardiac medication. The documented biological activity is real, which means interactions with existing medications are a genuine consideration, not a boilerplate warning. Grow it if you can. Purchase it if you cannot yet. Know what it is either way. Priya Mehta strips those leaves from the branch before sunrise and does not think about the NIH. She does not think about the USDA or the patent filings or the soy protein lobby. And she thinks about feeding her family. She has thought about that every morning of her adult life, as did her grandmother, as did her grandmother's grandmother. Across a continuous chain of practical, embodied knowledge that Western agricultural science spent the better part of a century classifying as non-commercial. The gap between her morning branch and a clinical trial is not a gap in botanical knowledge. The knowledge was always there. The tree was always there. What was missing was funding. And funding, as the patent record shows quite clearly, follows profit. Not need, not efficacy, not the 1,300 peer-reviewed studies sitting indexed and available and almost entirely unread by the people who need them most. What changes when you know this is not the tree? The tree is unchanged. What changes is the map you use to read the world you are already living in. Every food choice that costs more than it should. Every prescription that treats a mechanism a plant already addresses. Every municipal water system that chlorinates instead of coagulates. Those are not accidents of agricultural history. They are the compounded result of documented decisions made by specific institutions for specific financial reasons, at specific moments in the last 80 years. Some of which are named in this file. You now have the file. Share this with someone who is paying for something this tree does for free. Because that person exists in your contacts list right now. Not as a favor to an algorithm. As an act of translation, moving documented knowledge from one world into another where it is needed and has been kept from arriving for too long. If this reached you, subscribe. Because the next file in this vault describes a flowering vine the EPA classified as an invasive weed in 1978 that was used as a frontline wound closure treatment by military field surgeons on 3 continents before being formally removed from battlefield medical supply kits in 1962. And that is currently the subject of active wound healing regeneration trials at the University of Edinburgh, trials whose preliminary findings have not made a single mainstream nutrition or medical news outlet.